Welcome to NewYorkBIO's Virtual Breakfast Series, a digital program started in 2020, bringing you fireside chats with leaders from across the healthcare spectrum. This week's episode features Chris Mason, a serial entrepreneur and professor at Weill Cornell Medical School.
Good morning. My name is Jennifer Hawks-Bland, and I'm the CEO at NewYorkBIO. We're thrilled to welcome you to another installment of our virtual breakfast series. This week, Dr. Chris Mason is our guest. And special thanks to VWR for sponsoring our virtual breakfast series for the months of November and December. It's really been fun. I know that a number of you have told us over the last few months that you were surprised at how much you enjoyed dropping in for a NewYorkBIO hour each week. So we have been it's been a lot of fun to do these and have really interesting conversations with folks from all across our ecosystem. And this week is no different. We have an amazing guest in Dr. Chris Mason, and I am going to kick it over to Derek to introduce him, and then we'll start our conversation. Oh, and one last housekeeping measure for those of you who might not be familiar. You can ask questions throughout our discussion via the chat or Q&A feature, and we will lob those into Chris.
All right, so Chris, good morning.
Good morning.
We are thrilled to have you, and it's rare that we get to talk to somebody that has kind of spread out so broadly over a scientific area like genomics, where you've kind of covered literally everything from space to the subway.
That's right?
And we've got a lot that we can cover today. And what we generally like to start with is a bit of an origin story. And I thought with you, I thought it'd be really interesting to ask you kind of how you got to New York, right? So you came from Wisconsin, you did a postdoc at Yale. And what was kind of the process that led you to Cornell? How did you get there, and what were your dreams when you first thinking about it?
Yeah, great question. So I think originally born and raised in Wisconsin, did my undergrad there. So I still have some Midwestern wholesomeness that's buried deep down inside, which is now mostly covered by some broken glass and tar and impatience of a New Yorker. So, but it's still
glitter.
in there.
That's glitter.
Yeah, that's right. That's right. Glitter, the glitter you can't get rid of, right? The herpes of crafts, So it is, it's still in there.
Thank you.
But I was at Yale and actually did some,
series.
Metro North to
Join
come
us
visit
every
people at
Tuesday
Columbia,
at 9 a.m.
and
for
Cornell
more
back when I was
discussions
doing my
with
PhD.
leaders from across the
And then,
healthcare spectrum.
in my
For more
postdoc,
information on New York
I actually
Bio,
moved to New
please
York,
visit us at
or was looking in New York City in related areas like Cold Spring
www.newyorkbio.org.
Harbor, when I started looking at potential places for, the faculty positions, and it was for a girl. So as many things in life, it's like, well, I moved for a girl. So it was really that simple. My then girlfriend, now wife, Joan, was living in New York. She had just moved there when I was almost done with grad school. And
what was the right choice then?
Yeah.
On all levels.
Well done.
Yeah, 12 years later, still doing great. So, but then, looking in New York City, there's, of course, as you all know, there's a lot of universities, a lot of great biotech. It's a great place to look around. And so looked at Columbia, looked at NYU and Cornell, also looked at the NIH as a position that was there. I was fortunate to get offers from all places, and then really just had to decide where did I think some of the most cutting edge science and most, some of the interesting people. There's great people all over the place, but I just had a really great camaraderie with some of the people. There's an Institute for Computational Biomedicine, so it blends a lot of the cutting edge methods in molecular technologies and biology with computational methods and algorithms and a lot of machine learning approaches that 10 years ago were actually very, very new, being applied to biology. Some of the methods themselves are actually very old, like machine learning is decades old, but it's all exciting now, and people just run a regression and they say they're doing machine learning. But but basically that, like, where was the home where I felt like I could get the most science done and go the furthest in terms of ideas. So and then started in 2009.
Wow. Okay, so I need to ask you, when you were giving your job talk, the tagline on your lab is now a 10-phase, 500-year plan for the survival of the human species on Earth, in space, and in other planets.
Yes.
Now, did you use that in your job talk, or has that evolved since then?
So I did. I confess that was not my opening line for my job talk. It would fit well in some places, but not in a normal sort of stuffy academic room. So
yeah,
certainly not at a medical school. So I did talk about it. It was it was a lot of the ideas behind that were public, were things I would discuss, of course, like at the faculty dinner, during the interview dinner. That's a good time for that conversation, but not sort of the opening line.
Right.
But it does beg the question, like, why do people have any long-term projects, or how do you decide what you're going to spend your life on? And a lot of it was just unraveling some of the missing components of the human genome at the time. Like, I really focused, my training a lot was in human genetics, and I really, there's still so much of the 3 billion letters we don't understand. I'm like pleasantly haunted by the fact that we don't know what all of them do. And it actually was the main genesis of me getting involved in genetics, is that, you just start as one cell, all the instructions are there for every other cell. And it's just a question of teasing out what each letter does, and how it keeps getting modified. So as you develop. So that was most of my pitch. And then, as a geneticist, but then I said, well, what you could do over time is, if you learn that well enough, you could actually start to think about modifying the genome, or strengthening it, or, repairing it if it gets broken, as we try to do with immunotherapies. Basically, we have, something goes wrong, you design a cell to go after the aberrant ones. And so some of these ideas of engineering biology, which have been in the field for a long time, become much more exquisitely refined as we map the human genome and improve tools. And then, of course, started to look into other genomes, not just human genome. But that was the original pitch, was focused on the human side. Yeah.
Yeah, it's interesting. The more we tend to learn, we tend to learn more about what we don't know.
Yep.
And then we learn that our tools aren't good enough, and then we make even better tools, and then we learn that the stuff we thought we knew wasn't quite what we thought it was, and then there's even this other stuff that we still don't know.
Yeah.
So
it's basically job security. If we already knew everything there was to know, we'd have nothing else.
I found this great thing, but I have to go look at all of this other stuff, because it turns out we really don't understand it. Yes. So what were some of kind of the big things early on that allowed you to spread out a little bit, or to kind of go down a few different paths?
Yeah, I think it's like starting a lab is a lot like starting a business. I think a lot of your audience is in the entrepreneurial space, and people think, okay, you get a pile of cash, and they say, okay, here's some money. It's in starting lab, and faculty positions are very much like this. They say, here's a pile of cash. Usually it's $1.5 or $2 million, which is actually, like, that's a lot of money to get started when you're just one person. But then with that, you have to buy all your equipment, get, hire all your people, and not just hire them for 1 year, but you probably need them for maybe 2 or 3 years, right? So suddenly you do a budget projection, and you think, well, I need to start writing more grants and getting more funding. Otherwise, I'm going to run out of money pretty fast. And so, because I think, as you all know, some sequencers at the time, like one of the big bets we made was on the PacBio sequencer, which was $700,000 for one piece of equipment, right? So, and then, service contract and other components. So we made a lot of big early bets on technologies that were really bleeding edge. But that, I think paid off, because we could peer into biology in ways we couldn't before. So in that case, with Pacific Biosciences, it's a sequencer that lets you, of course, sequence human genomes, or any genome.
Yeah.
But you also get this kinetic information as the polymerase works. So you can actually see, is that base normal cytosine, or is it methylated? Is it modified? So you get both genetic information and epigenetic information simultaneously, from a paper Eat It All in 2010 that first showed it. And so we thought, well, this is pretty powerful, because you can get multiple modalities of the biology every time you run the instrument on any sample. Now, it took a bit longer for it to get, perfected and optimized, and the algorithms for calling it took, longer than anyone wanted. But now it's actually, the stock has almost never been higher. It took, a long gestational period after its IPO to really get mature, but now it's, one of the best sequences in the market in terms of just overall accuracy, when you look at what the machine puts out. So Illumina is the one that everyone thinks of, but PacBio is arguably the most accurate sequencer on the market.
Well, they also have it's a long read sequencer too, right? So that gets rid of a lot of the structural variation challenges that Illumina has.
Because then you can see them, and so it has, so one of the things we did is, I put in funding, and the department put in funding, and we got, kind of passed a hat around to try and get enough funds to get it and start to do experiments with it. So we published a lot on human genome sequencing, bacterial sequencing. So that was an early bet. It was to, look at new technologies, in that case, sequencing-based technology, bring them into the lab, start to work with them, get them optimized, and then make it so that we could actually learn new things about biology, which makes it easier to publish papers, makes it easier to get new grants, and say, we have a way to see something that no one or very few people are able to see. And so become a bit of a technophile as a scientist, because you can never marry technology because it always changes, but to be on the cutting or bleeding edge lets you often peer into a window of biology that you couldn't before.
Right.
You're creating a flywheel, right, that would sustain additional growth and new avenues to
Yeah.
look at.
Exactly.
Yeah. How do you learn new things? How do you ask better questions? And then once you answer those questions, how do you kind of stay two steps ahead of everything else?
Yeah, exactly. Yep.
So okay. I think we have two places to go. We can either go to the subway or we can go to space. But I think we're gonna start with space.
Okay.
Right. So
Great.
I think I think it was about 2014 or so, right, where they had put together this consortium to sequence folks on the International Space Station, and a little while after that came the kind of the landmark twin study, right? So Mark and Scott Kelly were on the ISS, and Cornell was part of the team that did that and showed that if you spend longer times in space, actually weird things happen to your genome.
Yes. Yeah.
So how did that even start? How did it come about? How did you get on that team? Just where did that come from?
Right. So we'll take the genesis story there. So, and as most people probably know, Mark Kelly just got elected Senate in Arizona. So now Senator Kelly, and we've been banking blood samples of them since 2014. Just got new ones a few months ago. So we now have sort of the first senatorial genome project starting.
Do you have the ability to tell whether the election changed his genome or not? You do still have the control samples. I mean, this is something that you could.
Yeah, I was just emailing him like epigenetic changes, stress-induced changes.
Yes, definitely.
What will a year in the Senate do to this poor man's genome and epigenome?
Yeah, I don't know if Nature would take that data, but I'm pretty sure the evening news would pick up on it.
Yeah,
You would get airtime.
So, so that actually that brings back to the genesis of the lab. Is that when I realized, started doing budget projections, said, oh crap, I need to get more grants. You can send to NASA's like anything. It's like the NIH or the NSF or other funding agencies.
Right.
You can send them new proposals, or you can respond to an existing request for applications or RFA, where they say, we have an idea, we'd like people to give us your best thoughts, and we'll pick the top 10 or 20% and fund those efforts. Or you can send them, say, I have an idea and I would like to do this with NASA. So I actually sent them an unsolicited proposal in 2011 that said, I think we should do genome and metagenome and epigenome of astronauts and see what happens when they're in space. And it turns out they had, it's interesting, one of the actual program director of the human research program at NASA, John Charles, who emailed me back, he said, we actually don't have bank samples, like viably frozen cells, to even answer some of the questions in your proposal. And he said, but it was really interesting. Keep tabs on this page and we'll keep in touch. He said, I think there's plans within NASA to start to do more of this, but NASA had never done, genome sequencing, RNA sequencing, looking at metabolomic profiling, any of the sort of cutting edge work in epigenetics. Most of it was because they didn't have an IRB in place to do human subjects research for some of those questions. Like if you sequence the genome, it's like the movie Gattaca. Would you be able to tell who should go to space and who shouldn't? And they had no framework, and actually still don't plan to anytime soon, of how do we pick genetically which astronaut should go. It's far too complicated a question. We frankly don't know enough about biology, or the pleiotropy, which is the one gene having many functions problem, in biology. But so they said, well, hang in there, and there'll be something I think you'll like soon. So then they made an announcement that said, okay, we have this unique twin study. This is a couple of years later. That is, we're requesting ideas for grants. If you could study two twins, one in space for a year, send us your ideas. So it was great. As I already had the whole grant written. Right? I'd already written. So all I had to do was kind of update it, put the latest papers in there, and I was like, okay, great. So, and you never know if you'll still get selected, but we then were one of the 10 teams that were selected. So it was kind of like a Michael Crichton novel, like in Jurassic Park, where they had like a mathematician, a biologist, they had an ecologist, they had all these different people coming together to go to that island. In this case, we tried to cover the panoply of all of human physiology and cognition and behavior. So there was a cognitive expert, there was a sort of a DNA and RNA expert, geneticist myself, other people who'd worked a lot with microbiome. We later did a lot more with the microbiome, but people working a lot on circadian rhythm. There were people doing metabolomic profiling, data integration, statistics. Like it was really a good team of like all the 10 teams and all the co-investigators. So we got together and had our first meeting, and flew to Houston. It's actually the first and only grant. It might be the only one I ever will get. What they said when they send you an email like your grant's been selected. We'd like to let we're letting your congressman know that you've been selected. I'm like, why is that happening? So that was really
okay.
Okay, thanks. I never received anything from the congressman at the time. But But so it was really kind of a cool opportunity. And then, then then we realized, okay, they announced the mission we had to do, but they had no protocols for how do you get blood in space and get good DNA and RNA and protein.
I was wondering about that. Yeah.
So it was like a crash course of, okay, what tubes are approved for transit to the space station? Because you generally don't want anything with glass because it could break and shatter and get in the eyes of the astronaut.
Everywhere,
So
right.
we had to build off, most of it was already approved and also set up for flight. We also had to prepare for what's called ambient blood draw. So blood gets drawn in flight and then put in the Soyuz capsule, brought back to Earth, and then sequenced. And so we sometimes got samples that only 35 hours prior had been, orbiting the planet, and we could get them in our hands in Houston.
That's so cool.
Really quickly. So we had to get protocols for that, and also do, controls as well on Earth to see if you want to make sure you don't just measure cells dying on their way to you. So we had to control for that as best we could.
Right.
And then bank samples. So measured everything we could. So immune cell populations, whole blood fractions, microbiome, looked at all the RNA changes, microRNAs. We actually have, it'll be next Wednesday, there'll be a big press release from Cell and NASA on a bunch of follow-up papers, which will be on the cover of Cell and Cell Reports and iScience all at the same time. It's actually, we got 3 scientific journals that feature on their cover pictures that Scott Kelly took when he was in space for that year-long mission. And it's all these different follow-up studies that we just finished in the past year. So those will all come out next week.
That's awesome.
That's
so it was a lot of effort, but it was really great in collaboration with NASA. Like, I don't have a helicopter that's in Kazakhstan that's going to bring me samples, but they do, right? So it had to be collaborative to get samples repatriated and brought back. And we found interesting things. So he, actually got his telomeres got longer in space. So some of the measures of aging look a little bit better, but it went away when he got back to Earth. We've now
interesting.
Confirmed this in 11 other astronauts, and that's what's coming out next week. Is that it seems to be a consistent feature of spaceflight. He didn't get epigenetically that much younger or older. He stayed about the same, so that doesn't seem to be driving too much of the aging in that direction.
What time period are we talking about here?
2015 to 16.
Okay, so it's about a year.
Yeah, he's 340 days technically, so not quite.
Okay.
A lot of people will give him crap and be like, you weren't in space for years. Like, it's 340 days in consecutive days. Like, it's pretty close.
It's close.
Yeah,
we'll give it to him.
Yeah. And then also a lot of his physiological changes, like bone loss and muscle atrophy, you can see when he landed, his body really had this pro and anti-inflammatory battle. Whereas trying to respond to, like, basically the muscle saying, I'm now having to deal with gravity. How do I handle this? His body broke out in a rash everywhere he basically had swollen feet. He said his ankles swelled up to the size of almost basketballs. So he really had some painful response to, being back in gravity after a year.
Yeah.
But most of it went away. What's interesting is when we looked at his body's biochemical profile, IL-6 is interleukin 6 is a molecule that's also upregulated in COVID patients.
Mm-hmm.
So when we were looking at the data, actually COVID data earlier this year, there was this weird moment. I was texting with a postdoc in the lab, or now senior staff scientist, actually, Jem Maiden, and we were like, well, it seems like we're looking at the NASA data again, but we're looking at COVID patients, just because the stress on the body is akin to being in space for a year and then suddenly having to deal with gravity. It's in terms of that, at least that molecule resembles that in the body.
All right. Well, we'll get to COVID in a sec, but I wanted to just ask you, what are, in addition to maybe some of the inflammatory things, what are some of the kind of broad human health either lessons or things that you take out of a study that's done in space, right? How do you equate something that most people will probably never experience unless we have mass colonization exodus to human health here?
Yeah, I mean, so this is obviously just phase 1 of the 500-year plan. So this will be more relevant for people that are 30 or 40 years now. But what you can do today, there are some things today. So some of it is the technologies. We did sequencing in space for the first time. So getting algorithms and portable supplies and protocols that work anywhere is helpful for remote diagnostics.
Sure.
Learning about these signatures. Now we know we've compared them also to to twins who climb Mount Everest, and we see some of the same signatures. We see it in COVID patients. So some of this is learning about how does the body deal just in general with extreme stress. We're interestingly starting to see, some consistent features. And so that means that some of the pharmacological interventions that are used for astronauts or patients for COVID or vice versa, that can inform each other in terms of what pro and anti-inflammatory molecules can you sort of ramp down or just control, because, as you've all probably seen in the news, it's not necessarily that your immune system responds to COVID that kills you, but it's that it over responds, and say neutrophils, macrophages completely swarm the lungs and lead to so much inflammation. That's part of what kills you. And so, understanding those mechanisms can be helpful for other diseases. We've already seen that.
Sure. I mean, it's relevant, everywhere. When you think about oncology, you think about cell therapies and CAR T. One of the biggest challenges was cytokine release syndrome or cytokine storms, which is effectively the same exact thing,
Yep.
right? And controlling the body's inflammatory reactions is really kind of pretty key to either tuning it up or tuning it down, or getting it to get the effect that you want.
It needs
So
to
Yeah.
be
okay,
Goldilocks.
Yeah.
we're gonna come back down to earth, and we're gonna go and we're gonna go
Well,
under the ground and into the subway.
I'll
And
put a
this is
quick link. We just published a paper
go
this
for
year
it.
on like on controlling cytokine release syndrome for CAR T therapy. So I'll put a quick paper there that we just published this
Okay.
year.
Oh, awesome.
To
Yeah,
your
I
point,
can.
like it is a key question. If you do it there, it's help for immuno-oncology. Absolutely. Yeah.
Mhm.
Yeah, no, I'll post this on our Twitter and LinkedIn pages after here as well. So all right, so let's come back down to earth. Let's go into the ground and let's talk about the subway, because
Okay.
one of the I think more famous things that you've done is also the subway sequencing project, which started as PathoMap,
Yep.
sequencing all of the New York City subways in one waterway, if I remember correctly.
Yes, the Gowanus
And
Canal, which by the way just started dredging this morning, actually. So they're finally cleaning up, which I'm a little bit sad about. So we've been sampling it every year to keep track of how it'll go. Yeah, but yep, the Gowanus Canal.
you can sample it getting cleaner. Cleaner,
Yeah,
cleaner
that's
is
right.
a
That's
good
what we're
thing.
gonna
You,
do.
you're
Yeah,
just you're just sampling change. The
that's right.
data's going to change, but you can still monitor it. So where did that come from? Because honestly, I think the germs on the subway has been, you can think about the germs on the subway as a topic for all time in New York, right? So how did you get the idea to do this?
Yeah. Yeah. So it came from two places actually. So it was, as a human geneticist, it was most of my training and doing a lot of work in inherited disease, cancer genetics and epigenetics. And we've been sequencing human genomes for a while. And one thing that bothered me is that if you get, say, a million fragments of DNA from like from even a blood sample, but usually an oral sample,
Mhm.
most of the DNA is human because it's from a human body, right? But then somewhere between 1 and 5, sometimes 10% of it would be non-human DNA. You'd say, well, that makes sense if it's from your mouth, but sometimes you could even see it when you get it from like blood samples. And
Hmm.
so some of this is because the human genome was incomplete, and I have been and still am working on that problem with many others.
Mhm.
But some of it's also because some of those fragments aren't human, right? So I always just feel like every molecule has a home. Like if I see a fragment of DNA or RNA, it came from something. I would
Right.
just know where it came from, right? So it
Right.
nagged at me for a couple of years. Like, well, we have all these other fragments. What if they're like, you think of like HPV and cervical cancer? Like, in some cases, viruses can even have a real strong implication for cancer, or HCV and
Right.
liver cancer, the inflammation, Epstein-Barr virus. There's plenty of places where viruses can lead to or directly cause cancer. So, I knew all that, but it's kind of nagging at me. But then the thing that really pushed it over the edge was my daughter, who was at the time 3 years old, got old enough to like start, playing and riding on the subway with us on our way
Oh
in.
God, yes.
Yeah, and
Touch,
actually
touch,
did.
touch,
She
touch,
did
touch.
touch, put her hand in her mouth. One day she actually licked a subway pole,
Oh
like,
yeah, my
yeah.
kids have done that.
Yep.
And not because, just because it's, she's exploring the world with her mouth, which is, what kids do, and then what, middle school people do when they're dating, so
Yeah.
it's something you do at different times in your life. And it just really, I just, yeah, went home and I thought, what is on the surface? Clearly a microbial exchange had occurred. I wanted to know what it was. And there was literally almost no data whatsoever on what's on the transit system. There'd been a couple papers about buses in Portugal, one about the air in New York City, actually. That was by Norman Pace, who published a paper saying, it looks like the air inside the subway and the bacterial levels about the same as outside, so it looked like they were similar. He did a project with the MTA on that and said, well, they're about the same, but with an older technology. It's called 16S sequencing, which didn't
Hmm.
really give you resolution of other organisms or even species or even genus level sometimes. So we thought, okay, well, there's really not that much that's done. And contacted the MTA, started asking around, said, well, let's just start doing some pilot sampling and see what's there. And we found all kinds of things, because every time you swab a surface and sequence it, it's as diverse as the universe in which we live, or the world and planet we live in.
Yeah.
It's covered in spores. It's got bacterial fragments, viral fragments, phages. It's got human DNA, RNA, plant RNA, fungi. Everything is there. And I just wanted to know what it was, right? So we had a bunch of summer students who were coming in 2013. And it was like Cornell undergraduates and CUNY students, and some from Hunter College. And like,
Yep.
well, Dr. Mason, I've learned, your work in human genetics and all this other work, and I'd like to come and work in your lab for the summer. So I'd already accepted them and had 5 students. And then when they all got there, I said, I actually did this the morning before they got there. It was a bit dramatic. It was a bit overly dramatic, actually. But I said, listen, I like took a projector that had the New York City transit map on it and projected it onto this giant whiteboard and like traced out the subway system on this huge whiteboard. And they came in, and I was chatting with them and saying, okay, listen, human disease and cancer and these things aren't going anywhere. They're going to be here for a long time, and we're working on them. And that's all fine and dandy. I said, but what's never been done is building a genetic map of a whole city. How do you feel about going out and swabbing the entire city all summer to build up a genetic profile of the metropolis? And I pointed to the whiteboard, and there was like the whole transit system. And I said, if we're going to do something, you got to do it right. We're not just going to do a few stations. Let's do every single subway station in triplicate and build it out right. And they were super enthused. They said, like, let's do it. Great. So they, signed up to go, ride the train all summer to collect enough samples, built up an app. We built a crowdfunding campaign, a metadata tracer, built a barcode scanner. So like all these, it was a really great group of students, some of which are still working in the lab today, actually.
Yeah.
So.
No, that's exciting.
They didn't run screaming when she sent them to the subway.
No. Well, but my favorite thing is the stories they came back with. Like, we've all ridden the subway, right? So there is a wealth and diversity of humanity that's down there. And like, one person thought that one of my students was cleaning the subway with like the Q-tip. Like, oh, you're cleaning. Thank you for cleaning. And she said, no, I'm not cleaning. I'm just collecting. Other people thought there was one person who's like at the end of the 2 line who always yelled out that we were spreading HIV on the subway. And so Abraham Afshiniku has a good story on that. It's like, and that person's always there, just always at that station. So, there's just people who kind of live in different parts of the city, and you kind of meet them. So but the maybe more astonishing thing is most people who just didn't notice. You're like, oh, whatever, there's someone swabbing right there, I'm gonna keep walking.
Oh yeah, no, that's I don't think that reaches the level. I don't think that reaches the level that I would notice it. I certainly wouldn't say anything right now. That doesn't hit that threshold at all. Now when parents see their kids doing that, you can say, do you know what's on there? And they say no, and then you can just hand them the paper, and then they say, well, look, this is where we are, and this is what we're looking at.
Yep. And now, now it's actually very common to see people with gloves and masks and swabs in the subway.
Yeah.
There's actually a really great Instagram feed I highly recommend. I think it's on Twitter too. It's just called Subway Creatures, which is basically just footage of people who've caught weird things happening in the New York City subway. And it's phenomenal because we've all experienced weird things on the subway, and it kind of captures a lot of them.
It is a never-ending stream, I'm sure, of content.
My wife.
Go ahead. Good.
I was going to say, my wife and I just have a code word for stuff that we see that's weird in New York. We just turn to each other and say, where do we live again? Right? Because you shrug your shoulders and you say, yep, that's just New York City.
That's right. Yeah, even my daughter's now 10, and she'll do the same thing. We'll be walking by like a mattress on the street and like, a weird, TV. She's like, oh, it's New York City. There's a mattress on the street. Just and so now it's become a global. There's over 120 cities that all coordinate in the same study. So right after the paper came out, a bunch of people said, hey, what does Stockholm look like? Or, what does Paris look like? Or Hong Kong? And I said, well, let's find out. So we started a consortium that now does called MetaSub, which is the metagenomics and meta design of cities and urban biomes. So it's mostly about how do we map and design cities and learn from sort of their trajectories from a genomics perspective, which was fairly abstract until COVID hit. And then everyone said, well, maybe we kind of, turn the network to the attention to get all the RNA viruses captured across the cities as well.
Yeah. So thinking about COVID now, right? You now have, given all of this background in disease surveillance and in microbe surveillance, when COVID first, came here, and when it first started, getting bigger, what were some of the things that you were thinking about lab wise? What were some of the things you were thinking about sampling wise? And, what were some of the things that you did initially?
Yeah, we really wanted to see, because there is a question too, like became much more relevant when we went to the international scale, but even just thinking like, we wanted high touch surfaces, places that there's a lot of human interaction, like curbal interaction. So we went for turnstiles, the kiosks, and the benches, which are the core locations. But in the first study, we did look all over the place. We did some like store railings like before you get into the subway, the floors. We did some ceilings. So we've done some pilot work where we look at where if you look elsewhere in the train, what else can you find? And so most of it was pretty similar though. So we wanted to get to places that it's the most human interaction. One of my favorite things of the paper actually was that we could reconstruct the neighborhood in which you lived based on human DNA, because it actually looked like the census data. So you could say is it black, white, or Asian or Hispanic, different areas of the city. that itself is reflected in the DNA that's left behind. So if you could, characterize it, you'd say, oh, this looks like a Hispanic neighborhood, and it usually is actually. So it wasn't a perfect correlation, but on
Yeah.
average, if you see that, it's likely, you're in
It's pretty
Harlem, or you
good.
can see you're in Chinatown, you could actually see it from the human DNA left behind.
Wow. Right. So have you been active with things like New York City and things like that in terms of their testing strategy? So how did that kind of stuff unfold, and really how did your thinking unfold from kind of the very beginning into when we had learned more about the virus after it had been sequenced, and even after people had started developing tests?
Yep. So we worked with the MTA from some of the very first PathMap studies, but the
Mhm.
surveillance questions have kind of been, we're still doing that effort, but a lot of our effort on testing has been with something called the Pandemic Response Lab. I don't know if you've seen that yet.
Yeah.
Yeah, so just prl.nyc, and that came out of support with also the mayor's office and a centralized testing facility at the Alexandria Center.
Mhm.
Yep.
And so, we've been doing a lot of testing at Cornell. Obviously, we're helping a lot with our own backyard at New York Presbyterian and Cornell, and helping as much as we can with looking at phylogenetic differences of the viruses, the host response. We have multiple papers that have been published and several in review on what happens to the body when you get COVID. So that's all been ongoing. But the pandemic response lab was kind of a really centralized effort for the city to get as much COVID testing as possible. And I think, what's interesting is the business plan for that, because I think a lot of your audience has business plans, they pop up, is with new, like, well, we need to do this for like a year. So it's not like a normal business plan. They put all this hardware and
Right.
infrastructure for COVID with the hope that they wouldn't be needed in a year from now,
Yeah.
and the vaccines, which now looks like they'll hopefully be happening by March or April. We start to get distribution of the Pfizer and
Yeah.
Moderna vaccine. So things are
Right.
looking very good. But that's what I've been helping with there a lot, is really our own testing, some of our own development on LAMP, which is a point of care and rapid testing method, which I
Hmm.
actually recently got a mug that shows this. The New England Biolabs made these mugs of like 30 minutes. You can see a test. It's about the dorkiest possible mug, but why not?
Hey,
There are points for dorky mugs on this
That's right.
show. This
Right, right.
is you. That's a definite feather in your cap for this.
Yeah.
So, go ahead. Sorry.
No, so those are I'll put it in the chat box as well, but that's the place where we spent a lot of our effort. And also just trying to start to look at what happens to the body as you get COVID. One of the most surprising things is COVID was first treated just, as if it was flu when it first came through, because people didn't quite know what it was. We didn't, of
right.
course, know. And it's really interesting. Like if you look at some of the lung samples that came in from the COVID patients, both in the spring and now, you look at the lung tissue, should have alveoli, should be kind of porous. It's your lungs is a site of gas exchange, of course, right? So
Yeah.
it should be real open, but then some of them look like they were liver tissue, so compressed organ. Like with Rob Schwartz, a pathologist we've been chatting about and publishing on this, is it looks like it's the wrong tissue. It's like the lung has become like a liver in terms of its density,
Yeah.
and all these immune cells infiltrate and really lead to damage and inflammation. And so it's really a peculiar disease. The other thing we've seen is, as you've probably read about, is cardiac issues where we see damage to the heart. We see the virus enter, and some of the cardiac tissue also gets in some of the kidney, gets into the liver, lymph nodes. Like it's really, it's a unique virus and one we're still learning about, but every virus is different. So for example, it's easy to find COVID in stool, but it hasn't been seen yet in urine. But
Hmm.
Zika you can pick up in urine, but not as well in stool or other parts of the body. So,
Yeah.
it's kind of an interesting facet of biology. It's creepy and awful, but it's also intriguing, is that each virus has its own
Mhm.
tropism or its own place of the body it likes to go. And
Yeah.
that's part of the pathology. So you have to understand that to understand the disease. And that also depends on your diagnostic, right? So there's never been a flu test that's been confirmed for saliva by the FDA. So, but
Hmm.
we're working with Saliva Direct, Ann Wiley's team there. So we actually, where you can detect COVID from saliva quite well.
Yeah.
Yeah, and we had Prim Primseret, who runs
Oh, great.
Miramis, right?
Yeah, yeah, yeah. So
Yeah,
she's
she's on the New
she's
York Bio
demanding.
board, and we had her as a guest a few weeks ago and
Yeah,
talked
yeah.
about their pool testing.
Yep, yep, yep. They're working on, I know. And so, but there it's just, automation is the biggest challenge there. I know they're working on, but yeah,
Yeah.
yeah, great for sure.
Yeah. So also so the company Tempest actually kind of brought you on, Joel Dudley, who was originally at Mount Sinai, now a VP over at Tempest. So there was a press release that they brought you on to do some stuff with them with COVID as well.
Yep.
So how did that come about? What do they do? And what are you doing with them?
Yeah, so that became an opportunity, and Eric Lefkowski is the CEO there, and Joel Dudley. I've co-founded Longevity with him, and we've worked on a lot of other companies together. he went there as a CSO, so he left Mount Sinai to go on there to take on a full-time role as CSO. So he left behind like a well-funded, well-oiled machine of a lab and kind of saw a
Yeah.
big opportunity. And I can and then at some point when they were getting into infectious disease and looking at COVID, they'd seen all the work that we'd been doing, and then they really, I actually first had dinner with Eric Lefowski back in December and January a few times, and really, they were often making like, well, you should really come. Why don't you just come to Tempest? I was like, well, I have a whole lab in a nice place. I got, I can do my, if I want to do entrepreneurial things, I can do that too. I kind of have the best of both worlds. But then once COVID really hit, they said, no, we really need your help. Like, actually, can you come, like, in some way, take a leave, do something? Because, at the time, everyone was getting slammed. And it was bad in New York at the time. And then eventually, of course, it spread.
Yeah.
So I said, why don't you, take a sabbatical or short-term leave to come, help out as much as I could. So put in the paperwork and said, okay, let's do it. So we ramped up everything we could to do, two big things, which is COVID testing and then also COVID host response. So the testing is at the centralized lab. We're doing a lot of testing for the state of Illinois, about 15,000 samples per day. So running through, helping to get the FDA submission done, which got approved, getting everything in basically for the testing infrastructure. Also at home collection is the other place that will be coming out hopefully very soon. Also, working on the regulatory side, the technical side, the components of the genetics, and validating it with sequence-based methods. And then the second big area we're working on is, hey, man.
Hey.
Oh, yeah. I was just talking about you. Okay. Just take a, okay, just take it out. Yeah. Okay. So, the second thing is like, can we look at the COVID host response? So big, create a large infrastructure of samples where we know patients have been infected, learn about what drugs did they take, which drugs look like they work versus ones that didn't work, or can we, better understand a predictive capacity of what we can do for patients when they walk in the door.
Right.
And so building up both those things. So it's structured to be kind of just short-term leave, and so I'll be back at Cornell probably in January, February. It depends on how things go with the outbreak and everything. But I've basically just decided to do everything I can everywhere I go to help anyone with COVID-related projects for the at least until the spring. It looks like, hopefully.
It's funny when I read the so I knew that Joel had left, and as much as as much as it was a little bit sad because he ran just such a great group at Mount Sinai, I was excited for him to step into something that was really exciting. But I got to be honest, I read the press release that said you had stepped in. I'm like, oh no, I'm like, I can't take both of them. And because you read nowhere in the press release does it say that it's a leave of absence temporary thing. Right? The press release was very exciting, very top level thing. So I will admit that part of me was happy for Tempest and genuinely sad.
Even some of the deans at Cornell, like, wait, are you leaving? Is what's happening? So it did create confusion. But no, there was a Genome Web article that clarified some of that later.
I saw that. I checked your LinkedIn status. I tried to make sure that you weren't actually going anywhere.
Well, I mean,
they're only allowed to poach one Wisconsinian from New York City for 5 years, type of thing.
Yeah, and I a lot of people vote with their feet, right? So it's a really it's a huge enterprise. They're building a lot of amazing research directions. I wouldn't go if it wasn't someplace I felt could be transformative. Yeah, that's obviously why Joel left. But they're also expanding fast. They had 5 years ago they had 2 people, and now they have 1,600, and it's really ramped up quickly. And it started mostly with cancer and basically neuro and psych. But branching out into infectious disease and also integrating a lot of these. So, getting sometimes, infectious disease can change how you would respond to different cancer therapies. And that's,
sure.
So more and more it's becoming, it's like a really large scale precision medicine company.
Yeah.
But spanning multiple diseases. So it's everyone there's been great. It's really a fantastic place.
Yeah. Well, you mentioned before kind of having the best of both worlds, and I did want to talk a little bit about starting companies, because now I think you're on 5 now. Number 5.
Right. Yeah.
Okay. So what was the first one? So I want to kind of rewind a little bit and kind of go back to the beginning. What was the first one? What kind of made you wanted to start it as a company? Let's just rewind the clock a little bit and think about the first one.
Yeah, and the first one, I think, like every good entrepreneur, my first company it didn't work, but it was a really fun idea. So this was actually built on some work we did when I started as a postdoc. I did a fellowship at Yale Law School on genetic, basically mostly on genetics and law and how they intersect, and particularly on gene patents. So at the time,
Hmm.
a lot of everyone probably remembers you used to be able to patent genes, so no one else could look at that gene when you isolate it from any human sample,
Yeah.
including myriad genetics. So I filed a white paper on this in 2007, published a paper on it in 2008, and then started to meet people from the ACLU and other places saying it is kind of weird that you can't. How is it that genes are patented? So my fellowship at the law school was to focus on that. And then eventually we brought a case with ACLU and other patient advocacy groups and AMP, Association of Molecular Pathologists, to say, we think this is BS. We should be able, other people, if you want to sequence a gene to confirm a mutation, should be able to do so. Like if you want to get a second
Mm.
opinion for your X-ray in your tooth, that's no problem, right? That we say this is a medical need and also a question of sort of personal liberty. I just thought it was
Right.
weird that as soon as I take DNA out of my body, it's no longer my property. It just seems odd.
Yeah.
But there's case precedent that goes in both directions for 100 years, a lot of case law that is around this. So it from a statutory and even regulatory and legal precedent perspective, it wasn't crazy. It just no one ever really challenged it. So we brought suit, and then eventually it went through the appellate court, the district court. We won in the Supreme Court 9-0. And then after that decision, I was like, well, we should start a company so that anyone can sequence their BRCA1 gene and know their status. It's called Genome Liberty, was the name of the company.
I like
And
it.
like I wanted to like run down the streets and say, you get free testing, you get free testing, like Oprah, but of course wanted to make a business around it. But then the FDA started clamping down on 23andMe and any direct-to-consumer
Yeah.
testing at all. So
Yeah.
the landscape at that time was really bad. So actually, we started doing testing for some people, had a bunch of orders in, but then the FDA was really clamping down hard. I think with good reason for what was happening at that time. But so we ended up actually closing on the company, saying like this is just not the right time. But I don't have $2 million to spend on legal fees for the next 3 years, nor do I want to. Um,
Yes.
and also I didn't have the background at that time. I hadn't done like regulatory approval for 510K or FDA or EUA approvals.
Right.
Like I had never done that before. So as a young entrepreneur, I was like, I don't know what the hell I'm doing here. So we either need to hire people or raise funds or do both, right?
Yeah.
So that didn't pan out that well and close that down. But then
But you probably learned a lot, right?
oh yeah,
I mean,
yeah, for sure.
yeah.
Plus, like, how do you do a crowdfunding campaign, or how do you go talk
Yeah.
to your friends and family about, listen, I've got an idea. Can we do we did an event in Wisconsin actually,
Oh,
where
yeah.
I went and told people like, hey, we won the Supreme Court case. We think we should take this now to the next level. Do you want to sign up and buy a kit and get tested? So learning how to make kind of a pitch to a room full of people
Yeah.
to order. So it was definitely a learning experience. And then that led to, though, some of the work we started doing with the subway, and I started giving talks about that and met Nivo Hara, who was actually she was just about to look for a postdoc. Then we met at a conference, and I was looking at the Illumina Accelerator at the time, as well as other sort of places to incubate a company around doing shotgun sequencing for infectious disease. Because for the same reason, I want to sequence something and see everything that's there. I don't want to just look for 2 or 5 or 10 pathogens.
Right.
If you have an infection, you don't care what kingdom it comes from. You just want to know what it
Right.
is. So
What it is. Yeah.
that led to BioShow. It's went through the Cornell Runway program, which is actually, if anyone in the audience is thinking about like you don't know what to do if you want to do a postdoc, it's a great place. If you get selected, it's 2 years of funding to start a company. You get all this business training. So Niamh O'Hara, so we founded BioShow, which does basically infectious disease sequencing and AI algorithms to mine some of the k-mers and the sequence data to figure out exactly what pathogens there. We also have COVID tests, of course.
Hmm.
So that's there. Then also, and Joel and I had been talking about starting a company for a while. So longevity started. I'll just go through the list, I suppose. We want to just
Sure.
um, so and BioShow also is in the middle of a Series A. If anyone's interested in chatting about the raise, we're finishing closing out the round right now, and that's the company's been around for 5 years. Longevity started because Joel Dudley we talked about, and Paul Jacobson and I were chatting because Paul Jacobson is the CEO of Thorne, which is a probiotics and supplements company, and I met him. I was like, supplements kind of shady. What, and how do you know anything works? The probiotics, how
Right.
do these things stay? Does anything work? And he said, we know, some work based on the, physician reports and anecdotal evidence, but there's no centralized platform to really rigorously test all of our products. But how about we make one? What if we fund a new company that would do exactly that and bring as
Yeah.
much evidence-based medicine and clinical trials and testing to the product portfolio and see what really works. If something doesn't work, we should stop selling it.
Yeah.
I said, oh, it's interesting. So then we talked about, all of Thorne's portfolio, but then also longevity became not just testing that, but also measures of sort of gut microbiome changes, also aging. We've looked at, as you can see, different organs age differently based on your blood work as well as microbiome. So that's also launched as a product. Became a place to try clinical trials and really multi-omic data integration for for me, I'm more on the consumer side, like when people want to know before you get sick. Because I feel like once you get sick, there's this entire universe of companies and products for
Right.
you.
Right.
But what if we could actually do better monitoring before you get sick, or try and at least shift things? A lot of times it's called
Sure.
functional medicine. Most of the physicians in the network are functional medicine docs, which I hadn't met that many before, but they're basically the doctors who get the patients where no one knows what the hell's wrong with them, right? So they've had this diagnostic odyssey where no one can figure it out. And then you start to say, well, you've tried everything else. Before we do like surgery and take things out of you, let's try some probiotics. Let's try,
Right.
variations of
Mhm.
your diet. Let's try and see what that does. And but we wanted to build a platform that could actually do a readout and see if anything was working,
Mhm.
which we published a first clinical trial a few months ago and showed really good results there. So we're doing more of that now at Longevity.
Yeah. Medicine is very good when things follow dogma, right? You have X symptoms. This means you have Y, and you are going to take drug Z. Thank you
Right.
very much. Call us in 2 weeks. Let us know how you're doing.
Yes, yeah, and if it's not that, if it doesn't fit in that neat bucket, it gets lost all the times.
Right.
And the other term we knew, one is just is Bridgeomics, which is just a consulting company that does a lot of it's just Bridgeomics, basically boutique and mysterious consulting work that we do. And the other one is a manufacturing company that's called Direct MDX, which is trying to create a lot of these devices that run the LAMP assay that we've invented earlier this year and
Mhm.
Okay,
perfect.
cool.
Yeah, so it's more of a manufacturing company which just started, so it's in the early
Wow.
stage.
So how do you think about well, let's take a little bit of a step back. So when you were looking at starting Biosha and things, how are your original interactions with, say the investing environment within New York City?
It's interesting is New York always feels like it has to play catch up to San Francisco and Boston, and
Hmm.
so there is but overall it went well. It's just you can tell that I think the number of people you can approach in New York is not as big, not yet. I'm hoping
Right.
some that'll
Yeah.
change, but
That's
I
changed.
think that'll
Yeah.
get better, but I would totally agree with you on that.
and so I think so some of it was, oh, you should talk to so and so's in the Bay Area, you should talk to someone else who's in Boston. So yeah, we got a lot of referrals, but eventually, I mean, the investors, most of them came from New York or Boston, so we
Hmm.
did end up keeping a lot of it, and also Cornell invested, so we had we were able to keep it to a large degree in the family. But now we're doing the Series A, so I'm about to ask me again in a month, and I'll tell you how it's laying out. But I
Right.
think
Yeah.
I mean, about half the round is already filled out, but it is it's a mix again. I feel like investors are really enthused, but I feel like there's people often that have really big funds that are often on the West Coast who don't mind throwing 20 or 30 million dollars at an idea that's even early stage, and some people that will say, oh, my minimum check size is $100 million. I'm like, okay, well, I don't
Right.
need $100 million. So there are some people where the conversations are like, well, okay, I'll come back to you in 5 years or something, but
Right.
so it's a mix, but overall good. If anything, I'd say there are far more options today than there were 3 or 4 years ago.
Yeah.
Yeah,
There's
I would
also a
agree.
lot more incubator space, like the BioBat, some of the work that's at the J Labs place, the Alexandria Center,
Mhm.
so the Harlem Biospace, like all these places where you can actually go and have a place that is
Yeah.
IP reserved for your company. So that 5 years ago, I think none of those existed, right? So it's interesting to think that the
Yeah.
ecosystem is there, and
Yeah.
also NYU has an entrepreneurial engine. Cornell's working on sort of this, the runway program, obviously at
Yep.
Cornell Tech, but then also the med school is working on its own entrepreneurial sort of boosting and incubator. So ecosystem's far better, especially if you have a seed stage idea. It's great.
Yeah.
Plenty of places to go and places to fund it.
Yeah, I would tend to agree. I mean, we've been fortunate enough to we had our offices in launch labs for the last couple of years, and that team does an absolutely amazing job. And to see the impact that they and J Labs and Bio Labs have really had on not only giving the community a place to go, but honestly giving investors and other people that come to look at New York a place to kind of know where a bunch of these companies are sitting, right?
Yeah.
It kind of gives a little more structure and a little more of a home to where the companies are, because before you had to really look to find them. You had to figure out where they were, and like, oh, did these people's offices are there in a WeWork in Midtown, or these people have an office here, or these people have an office here. I think they helped not only provide a destination, but also provide a little bit more structure to the ecosystem, and made things a bunch easier.
Yeah.
And I can tell you just from seeing them day to day. I mean, the effort they put into kind of fostering that community is fantastic.
Yeah.
I think they're some of the best things that people have brought to the city.
Yeah, and I think but I really want. I feel like I want more, right? Because when you think about the amount of capital that's in New York City, and just also
Yeah.
it's in New York City, the infrastructure that's here, the like
Yep.
it is it's a great, you'd think it should be a hub, like it should rival or even surpass San Francisco in terms of a place like where it should happen. But often it is just logistical. Like there's not that much space, right? Because there's been development here for, 400 years, whereas in San Francisco it's been a lot less. So people don't mind driving for 45 minutes, but you're still just in Jersey, right? 45 minutes from there, it's still a lot of
Right.
place. So it's expensive here. That is one of the biggest challenges.
But
Yeah, I mean,
honestly, it's been interesting, right? And I've been talking to people in San Francisco, and as far as living, it's actually less expensive to live here than it
yeah,
is in San Francisco these days. But one thing that I do think will help is we have, as people and companies expand beyond Manhattan. I mean, Long Island City is poised to add a ton of life science space, this year and into next year and beyond. And I think that will help.
Yeah.
And so, and our job at New York Bio, right, is to help try and facilitate those things and those connections. So, yeah, so we've had fun, and been able to hopefully help some companies make connections there. But you're right. I mean, and when you look out and your investors are all over the place, those companies also, which have traditionally just invested within their own home space, are starting to look at companies in New York more. And I think once we have that cross pollination, it'll be helpful for everybody.
Yep,
yeah,
yep. And even just having where it all can, like I know the Economic Development Commission in New York City is working on this. Most of the universities are, the tech transfer offices, obviously NewYorkBIO's. I mean, having more and more places where you can go and say, hey, do you know, I've got a company around this idea, or it's a small molecule, or it's a new device, or it's a new AI platform. Who do you know? And so, yeah, at the end of the day, it's people and it's a network, and it's saying, oh, I just got the perfect person you should meet. So
yeah.
more things like this are great.
Yeah, no, I like to think of it as network efficiency in some ways, right? The network is kind of getting the right people to the right other people to make things happen.
Yeah.
So we had a question from our audience about a role for genetic testing to identify people who are at increased risk for severe responses to COVID. As we're learning a little bit more here, do you see a role for that kind of genetic testing, either with COVID or even any other what I'll call run of the mill diseases or indications? Can we do anything to figure out who's really at risk for very severe responses to things?
Yeah, so there are some, finally, it took a while to get enough data to start to really ask this question, but there is an a human genome covert research group that's called the integrative, it's the COVID human genome research group. It's international. It has actually their website. It's called COVID HG. I can put it in the chat box here.
Awesome.
But it is, basically, a large research group that's trying to, let me see if I can pull this up here. Just, compare essentially, let's see, they do have HGI, Mark Daly. So it's run by Mark Daly, who's now in Helsinki, and it's just called the COVID-19 Human Genome Integrative Working Group. I'll put this back here. So I'll also, that's actually not that useful of a link. I'll get the full link. That's just the name of it. But while I'm describing that, there have been some loci in chromosome 3 and a few other spots that look like they are probably risk factors, as people are going into, I think they're, if they get COVID, they can look right at the genome or look even at the transcriptional response at the site of infection. You can predict, who's looks like to be at high or low risk. And so it's not perfect, but it is getting to the point where we can, you can start to make some of these predictions. And so
yeah,
there's two efforts here. I'll put the links for both of them here in the chat box.
I mean, to be clear, to do all this, do you need to basically, how deep do you need to sequence? What do you need to do to actually figure out this level of risk, right?
Yeah, then you need really thousands and thousands of people that did and didn't
Okay.
get COVID, or that ideally got COVID, and you can see who really either died or had a severe response, or people that were long haulers, or people that cleared the virus readily and had no follow-up symptoms. So
Yeah.
some of those efforts, though, are finally congealing enough data to answer those questions, and so there'll be some preprints about this coming out soon. And the other thing we've been looking at, and we have in one of our preprints online, it's in review now, is from, if you look at the RNA of how the body's responding to the infection, not just your innate risk, but then your sort of temporal risk at the time of infection.
Yeah.
There too, there's risk factors that we can stratify high and low risk patients. And so
Right.
a lot of them are in genes that you might think, like IFID3 or interferon stimulated genes, things that control interferon alpha and gamma response of how the body's basically just responding to the infection.
Mm-hmm.
And so, these are things that can tell us what your likely, risk is. The final thing is we looked also at medications for all the different, from the electronic medical records, what are people taking? How do we learn from were they at higher risk, lowest based on what they were taking? So that's also ongoing.
Yeah.
We've got a few minutes left here. So in the last little bit, I mean, all of this stuff points to between risk determination and stratification and things. I mean, should we really be looking at an era where we're doing a lot more either prospective testing or surveillance testing as a population to think about what kind of things we're at risk for and how to better manage, what kind of things we're treating ourselves for?
Yeah, like in terms of like home testing, you're thinking, or
Any kind of, I mean, really, what should the future look like, right? Should we get sequenced every year when we go for an annual physical? Should we be testing things at home? Like in 5 years, in 2 years, in 10 years, what should we be doing differently about how we test ourselves for our own health?
Yeah, I mean, there are many companies I'm either working with or started that are making home testing options. So I think there's going to be, Tempest is rolling out theirs in a matter of weeks. There's other ones that are doing that's for home collection, but there's also like home testing. Like you do everything at home and you get the
Yeah,
information. There's interesting compliance questions about like if you get positive, but you won't get paid if you're COVID positive, would you then hide the result? And so there are, we had this happen in Wisconsin where someone who was supposed to get overtime tested positive and he didn't want anyone to know because they wanted to get the overtime pay. Like, so there's real considerations about when you enable people to do testing at home. But, in the arc of the diagnostic universe, I would trend that way, is that you want to give more chances for more people to do testing.
yeah,
Because the risk of sort of people, like fraudulently denying something or hiding something is there, but I think far less of a risk and not as big as the benefit of people being able to keep themselves safe and others safe. So,
yeah,
so that is there, but I think it's not as big of a concern, at least to me. And so I think, that'll start happening. I think clonal hematopoiesis, or when you get mutations in your blood, we have a paper coming out about this next week with Mark and Scott Kelly. They have different mutations in their blood that you can see, even though they're identical twins. So they've changed genetically as they've aged. But you want to see, it's like a mole on your back. It's not a question of do we all have moles or weird parts of our body? We all do, but you just don't want that mole to get bigger, right? So I think the same thing
right.
is true with mutations in your blood, or even microbes in your gut, things that are there that are normal commensals. If they're fine, you're probably fine. But it's when you see severe aberrations or increases in mutated clones, things that seem to be perturbed that are in the wrong direction, you want to keep an eye on those. And, maybe it could be, you don't have to do anything. It could be like a sequencer attached to your toilet that gives you a report every month or something, or it could be,
Yeah,
it's a home kit you just swab and send it off and you just know it's being tracked, but they don't have to do anything. So I think there'll be more and more of that so we could intervene before disease comes.
yep,
Yeah.
yep. Well, that's fantastic. So we've, I know you have a thing to go to at 10. I want to thank you for being here. You've been a great guest, and I'll let Jennifer close us out. But listen, thank you so much for coming on today.
Pleasure.
Yeah.
Thanks for having me.
Thanks, Chris. I think we've had probably the widest ranging discussion with you, from space to the subway and then toilet. Throwing in the old,
Space to the subway to your toilet.
So thank you so much for being our guest. Thanks to VWR for being our sponsor. And we hope everybody has a great day. Take care.
Have a good one, everybody.
Take care, everyone. Thanks. Bye-bye.
Thank you for tuning in to New York Bio's virtual breakfast series. Join us every Tuesday at 9 a.m. for more discussions with leaders from across the healthcare spectrum. For more information on New York Bio, please visit us at www.newyorkbio.org.